All right, thanks for the invite, Hugo, and it's a delight to be here. So I'm a bit of a strange bird here. I'm not going to talk about a particular technology or a particular company. I'm going to talk about, I guess, the best job in the world that I landed about a year ago when I was invited to be the CEO and president of Yissum. Yissum is the technology transfer operation of Hebrew University. I'm not sure that Hebrew University needs much of an introduction. We've just celebrated 100 years to the cornerstone laying by folks that you may have heard of, like Albert Einstein and Sigmund Freud, who were the founders of Hebrew U 100 years ago. The university is the top ranked in Israel and continues to be ranked as a top 100 university in pretty much every global ranking to date. 7 Nobel Prizes and a Fields Medal in math just in the last decade. To Hebrew University faculty and alumni. And I think the numbers on the right are actually very telling. More than 1/3 of all doctoral students in Israel study at Hebrew U. We have 6 campuses, including Israel's only agricultural and food faculty, and more than 22,000 students, 1,000 researchers, about half of which are in the applied sciences. Yissum, which is the technology commercialization arm, the business arm of Hebrew University, was actually born as an innovative concept. It's the 3rd technology transfer company ever created 54 years ago. And by virtue of its agreement with the university, it is the sole owner of all intellectual property generated by faculty members and graduate students of Hebrew University. So Ziv had to leave, but Ziv mentioned a very dear person, Professor Amnon Shashua, who co-founded together with Ziv both Mobileye and OrCam. Professor Shashua is a full-time faculty member at Hebrew University. And both Mobileye and OrCam were hosted at Hebrew University for a long time. That's my last slide on Yissum and Hebrew U before I talk a little bit about precision medicine, just to give you a sense of the candy store that I get to run every day in my job. More than 10,000 issued patents, more than 900 licensed transactions, and more than 130 spin-out companies formed by Yissum. We actually run an equity portfolio of 189 equity positions that we have in multiple companies that we've received by virtue of either granting a license or participating in the founding of a company. We, as Hugo mentioned, have a few blockbuster products. Most universities are very proud of 1 or 2. We actually had $4 billion products. You have already heard of Mobileye enough, I think, for today. But here's my last pitch, Mobileye, Huji company. 2 blockbuster drugs, Doxil, invented by Professor Bernholtz, and Exelon for dementia, invented by Professor Martha Rosine Weinstock. And, as Hugo said, the cherry tomato. It's actually the most successful commercial product ever to come out of Hebrew University. Not big on royalties, but certainly commercially a big success. We also have over 100 active, multi-year, multi-million-dollar research collaborations with a lot of MNCs, a lot of multinational corporations. I could fill the screen here with those, but this is something that we are very proud of. Hebrew U is the only university in Israel that actually opens its gates and embraces industry. If you travel the roads within any one of our 6 campuses, you will see buildings of industry, Applied Materials, Merck, Intel. So we don't shy away from industry collaboration. We've always said that need-based innovation, industry-based innovation, is something that's germane to good science, whether it's basic science or applied science, it doesn't matter. The only importance is it has to be very, very good science. I wanted to talk about precision medicine or personalized medicine. We're kind of moving away from personalized medicine and focusing on precision medicine as a field that's really growing and growing in importance globally because of healthcare costs, because of the aging of the population, and because of a couple of data points that I'll show again. But this is the definition. It's a form of medicine that uses information about a person's genes, proteins, and environment to either prevent, diagnose, or treat disease. And I think this slide tells the entire story. This is a slide I borrowed from Statista. It's a statistics portal. This is the number of patients with no improvement after taking those top-selling drugs, Remicade, Humira, Enbrel, worldwide as of 2015 for every patient with improvements. For example, for every patient improved on Remicade, 3 will not improve. For every patient improved on Copaxone, 15 will not improve. For every patient improved on Nexium, 24 will not improve. Now, why is that? And the answer is, it's because we are different. All of us are very different, and we respond to drugs and medications and treatments very differently. And so the emergence of individualized, personalized, precise therapeutics has really skyrocketed in recent years. If you look at that data, this is the amount of personalized medicines by FDA categorization that were approved in 2005. Only 5% of approvals were personalized versus 2016. 27% of FDA approved therapeutics have a personalization element to it. 73% of oncology drugs in development today contain a personalized element to them. And the global biomarker market in 2015 was $24 billion alone. That's just the biomarker. It's not a therapeutic, it's just the ability, the empowering of a physician or any treatment professional to try and understand whether the treatment they're about to prescribe is going to be helpful or not helpful, and what's the treatment paradigm for this. I'm going to showcase literally one technology a minute. I can't do this as fast as John, but I can cover 3 in 3 minutes. I will just give you 3 very cool ideas about where personalized medicine is taking us. And we'll start with oncology, because we all kind of think oncology is synonymous with precision medicine. So many of you, I'm sure, are familiar with the fact that when a patient with a cancer is treated, many times the cancer will come back. And the reason the cancers come back is very much like antibiotic resistance. When we treat a cancer in the body using some sort of a systemic therapy, then most of the cancers is eliminated, but there are a few cancer cells that become sort of resistant or unresponsive to the cancer, and the cancer emerges. So the responding cells will essentially shrink, the non-responding cells continue to grow. And what you actually see on the whole tumor is that it starts shrinking, but then it grows. What Dr. Ofer Benny from our medical school developed is an in vitro, it's a chip assay that takes single cells or just a handful of cells from the patient biopsy, grows that in multi-well plates, allowing dozens of combination therapies to be tested on that patient tumor in an accelerated growth fashion. So within 3 weeks, she gets a readout on dozens of oncology therapeutics, aiming to find the one cocktail that would show her this image rather than this image or this image. So this is a kind of a service center that provides a rapid screening of a cocktail of therapies, be them molecular, biological, or others, that allow you to target a specific tumor rather than kind of cycle through potentially ineffective therapies. The second is very interesting. This is an antibiotic resistance. So I don't know how many of you have heard of bacteriophages, but bacteriophages are the enemies of bacteria. So just like viruses are our enemies, bacteriophages don't care about humans much. They don't do anything to humans. We're actually chock full of bacteriophages, but they really, really dislike bacteria. They kill bacteria. And for hundreds of years, we knew bacteriophages are the silver bullet in the fight against bacteria. Okay, they would attack bacteria and kill it. And in fact, antibiotic resistance, which is skyrocketing, is trying to now harness the power of bacteriophages to treat hard to treat antibiotic resistant infections. So the process, of course, is you isolate the bacteria from the patient, you evaluate multiple GMP produced bacteriophages, which you either synthesize or purify. You then train new phages if you need to train them to do other, to attack new bacteria. You prepare the IV infusions, you treat and you monitor the patient. And if you need to treat again because some antibiotic resistance developed, we do that. I wanted to give you one shocking slide because I'm sure no one's going to show you a shocking slide today. This is actually the least shocking that I could find on one of these. This is the story of a patient that was treated last month in our Jerusalem Bacteriophage Center, the second center in the world ever to now be approved to treat with bacteriophages, hard to treat antibiotic resistant patients. This is a patient that had an orthopedic implant done on the lower part of his leg. This caught infection, many of them do, and the infection started creeping up the bone, and he was slated for an amputation. He was going to lose his leg. That patient was treated by Dr. Ronen Chazan from our Faculty of Medicine and walked out to celebrate on his two legs to celebrate Eid al-Fitr 2 weeks after treatment with his family, completely bacteria-free. Okay, so this is personalized, this is precision medicine. Finally, a couple of weeks ago, we announced a collaboration with Grail, a huge unicorn on the west coast of California. This is in the field of early detection of cancer. All of the cells in our DNA all the DNA in our cells are the same. Then what's the difference between a skin cell and a heart cell? Well, the difference is not the DNA itself, but the kind of coating that the DNA has. So the different coating on the different genes essentially tell the cell whether they're going to be a lung cell, a liver cell, a beta cell for the pancreas, or a colon cell. Now, when you take a sample from a patient and you just look at their DNA is going to look the same. One of the things that, and this is my last slide, one of the things that Professor Dorr has developed is a non-invasive, simple blood test that looks at the coding of the DNA, the methylation of the DNA, allowing you to determine the origin of the abnormal cells that are circulating in your blood. So he can show here the difference between, for example, in a myocardial infarction patient, the difference between myocardial infarction and controls, just simply through a blood test. Same thing for breast cancer and controls, and it can actually track improvement after treatment. And in fact, one of the major uses for that is cancer unknown primary. So a lot of patients come in with cancer. You isolate, for example, a solid mass from the leg or from the liver, and you realize that's not the primary tumor. That's a metastasis that just resided there. And you're looking at the patient, you're looking at yourself and asking, where is this tumor hiding? Where is it coming from? Well, through a simple blood test, we are now able to essentially, just like we do CBC and urinalysis, Professor Dorr in collaboration with Grail are opening now a panel that I bet, in a few years will all be doing almost on a monthly or semi-annual basis. Simple blood tests will tell you whether the amount of lung-derived, pancreas-derived, etc. cells in your blood, are they within normal range, or something's going on, for example, with your kidney or with your endothelium, and be referred to treatment. So early detection, better treatment decision on chips, superior outcomes. This is what precision medicine is about, and I invite you to talk to us more, visit our website, because I have 350 technologies like that waiting to be partnered and commercialized.